Hello everybody. So today is going to be a little lecture, so it's a bit of an experiment for me as well as for you. So I'm just going to record an introduction to the terminology which is commonly used in the topic of cell division. So I know some of you were in class for this topic, but I appreciate that some of you must have missed it. And this terminology is really important because without it, it's kind of difficult to make sense of all the processes which are happening during cell division. So that's what we're going to do today. So we're looking at cell division terminology. Okay, so some of the concepts I hope are going to be familiar to you. So I hope you're going to recognize the picture I'm drawing here. So this is a cell. And obviously, I'm hopeful that a lot of you will be able to label some of the structures within this cell. So the outer line would be cell membrane. And the inner line right here would be nuclear membrane. Now, I hope after completing assignment 1, you do remember that inside nuclear membrane, inside our nucleus, what we find is DNA. And when cell is not dividing, this DNA is found in the form of really long, stringy lines, which are not actually visible, even though I'm drawing them there for you, but this is just a visual representation of those DNA molecules. And that sort of tangle of DNA molecules is called chromatin. So what chromatin is a lot of DNA molecules all tangled together in this big kind of mass. And when you look at a cell under microscope, all you really can see is a lot of sort of grainy dots, and you can't actually pick out individual DNA molecules because they are just too thin for the human eyes to see. So before a cell can divide, so prior to division, what needs to happen to this DNA needs to make a copy of itself, so it needs to be copied. And we call this process of copying of DNA replication. So copied or replicated, if you want to use proper terminology. Right, so replicated, you remember the process of replication was question, I believe, 2 in the first assignment. So I hope most of you are familiar with that term. So if I now take my tangle of chromatin and I pull out one of the molecules of DNA out of this tangle, so let me draw it like this here. Now, for the purpose of simplifying matters, I'm not going to draw this DNA molecule as a double helix because it's going to make my diagram quite complicated. So just keep in mind that that line, that squiggle, represents a double helix, double twist of DNA molecule. So this is a single DNA molecule. And so what needs to happen to this DNA molecule, as I mentioned here, it needs to be copied or replicated. So if I do that, I'm going to have one copy of DNA here, and I'm going to have my second copy of DNA on this side. Now, these two copies of DNA, they don't sort of just drift apart. They are initially connected together in this particular spot. And this spot, this little dot, which is usually on the picture shown as black circular dot, is called centromere. Centromere. So this is another bit of terminology which you need to be aware of. So centromere is simply holds two copies of DNA molecule together. Okay, so on a lot of pictures within assignment 2 cell division, you need to indicate where centromere is, so that picture shows you where it's found. Okay, so as I mentioned before, those two lines, those two kind of parts or shoulders of that structure are my two copies of DNA. So two copies of DNA. So that side, that line here, contains exactly the same information, the same genes as this line here, because they are identical, they are copies. So what happens next? Well, you know, we've done our two copies of DNA. The only problem at the moment is that those two molecules of DNA, they're really long and stringy. And so it would be really hard for your cell to, you know, move those two copies of DNA around the cell, because those long and stringy sections would just get tangled with each other, a bit like, you know, long and stringy bits of wool. So if I just, because I've run out of space here a little bit, so I'm going to redraw that picture here. So these are my two long stringy copies of DNA still connected by centromere. So what needs to happen next is that DNA needs to go through a process called condensation. So that's what I'm going to write here. So it's condensation, right? Condensation. So what happens in condensation is very simply you take your long and stringy molecule and you wind it around in this tight little package. So instead of that long stringy side, what I'm going to have is a really nicely tightly packed structure on this side, and my second copy of DNA is going to do exactly the same thing. So the whole thing is going to look a bit like that. You will still going to have centromere. So the centromere still stays where it was originally, and it's still holding my two sides, my two copies of DNA to each other. So this is my centromere. So that whole structure is then, once it's undergone condensation, this structure is known as chromosome. Right? And I hope you heard this term before. So this is chromosome. So those two sides, two copies of DNA, they are known as sister chromatids. So this is CH chromatids. Right, so two sister chromatids. So sister chromatids, they are always found on the same chromosome. So this whole thing, that whole structure is a single chromosome. And then the two sides, which originated from my two copies of DNA, they are sister chromatids. Okay? So just another bit of terminology, because this is something which does come up in the topic of meiosis, is if you have two chromosomes side by side. So let me just draw a couple of chromosomes. So let's say that's one chromosome and that's a different chromosome, right? So again, this is one copy of DNA and that's another copy of DNA. So these two sides are sister chromatids. So you see, I'm just repeating it from my label right here. So they're sister chromatids. These two sides are also going to be sister chromatids. But if I choose to look at two sides of two different chromosomes, I can't call them sister chromatids anymore. So they are known as non-sister chromatids. Right? So whoever was given the job of naming them. If you're surprised about this sound, this is just my cuckoo clock. So let's wait for a second for it to stop doing the cuckooing. And then I'm going to carry on. So they are known as non-sister chromatids. Okay? Okay, so the next question is, well, how many of these structures, of these chromosomes, do you find in a nucleus in a cell? Well, if you're talking about a normal cell, right, so something like your skin or your liver or your brain, etc., etc., etc. So just pick an organ you want. So those cells in humans, right, in humans, because different animals will have different number. So in humans, you have 46 chromosomes. Right, so 46 chromosomes in your normal cells. And that number, the 46, it's known as diploid number. So this is diploid. So a bit of terminology here, diploid number. So diploid number means that it's like a full number of chromosome, which you would find, as I said, in your bog standard normal cell in your body. Now those 46 chromosomes, they are arranged into pairs. So they're arranged into pairs. So why are they arranged into pairs? Well, because if I take a particular pair of chromosomes, so let me draw again a couple of chromosomes side by side. In each pair of chromosomes, one of them would have come from your mother, and the other chromosome in the same pair would have come from your father. Right? So when you were created originally, your mother contributed 23 chromosomes in an egg, and your father contributed 23 chromosomes in a sperm cell. So altogether, they made 46 chromosomes, or 23 pairs. So in each pair, you have one originating from the mother, one originating from the father. Now, those pairs of chromosomes, again, they have a specific name to them. They're called homologous chromosomes. Right? So what does that mean? It means that within each pair, those chromosomes code for the same sort of information. So let's say if that part of a chromosome codes maybe for your eye color on the chromosome which came from your mother, then the same section on your father's chromosome also would code for an eye color. I think I missed a letter here, there you go. So eye color. So if you take a different pair of chromosomes, of homologous chromosomes, they would code for their own selection of traits or features or characteristics in your body. Okay? So let's say I've taken a different pair of homologous chromosomes, so I'll draw another couple of chromosomes here. So that's another pair of homologous chromosomes. And let's say in this pair, this section codes for your ear shape, then the equivalent section on the other chromosome, on your father's chromosome, would also code for your ear shape. Okay? So let's say if I picked up this chromosome and that one and put them side by side, they will not make a pair of homologous chromosomes. So they're from two different pairs because they code for two different set of genes, for two different set of characteristics. Okay? So in that case, if you pick two different chromosomes for two different pairs, they are known as non-homologous chromosomes. I think the same guy who came up with non-sister chromatids, he was given the job of coming up with a name for non-homologous chromosomes. So it's not particularly inventive name, but it kind of describes what it needs to describe nevertheless. So let me just write it down. I'll go on to the next page here. So what I was talking about, if you have one of your pairs of chromosomes, so let's say this is pair 1, and then I've got a different pair of chromosomes, so this is pair 2. So these two are homologous chromosomes, these two are homologous chromosomes, but two chromosomes from two different pairs, right? I'm talking about the whole chromosomes, so don't get confused between sister and non-sister chromatids, because these are just sides of a single chromosome. So I'm talking about the whole thing, right? So this chromosome and this chromosome, they would be non-homologous chromosomes. Okay, so they're non-homologous. Two different pairs, therefore non-homologous chromosomes. Okay, so one other thing. So I mentioned that in a normal cell in your body, you have 46 chromosomes, or diploid number. You remember, I just remind you. So 46 means diploid number. Now, there are some cells in your body which do not contain 46 chromosomes. So I wonder if you know what these cells are. I kind of already gave you a hint, actually, a bit early on. And those exceptions are egg in a female and sperm cells in a male. Right? So in those two types of cells, also called sex cells, instead of 46 chromosomes, you have half that number. So half the number of chromosomes in a normal cell. So that makes it obviously 46 divided by 2, because that's what half is, you will have 23 chromosomes. So found in a sex cell. Again, in humans, right? So we're specifically talking about human body and human cells, because different species will have their own completely different number of chromosomes, okay? But in a human cell, 46, which is diploid number, which is in the normal cell, and 23 chromosomes in a sex cell. Now that number, 23, is known as haploid number. Okay, so diploid for the full number, haploid for the half of the number of chromosomes, which, as I said, only found in sex cells. Okay? So this is some of the terminology. Just one other bit of terminology which I wanted to mention as well. So if I go back to my 46 chromosomes, remember that was diploid, and as we discussed earlier on, those 46 chromosomes are found in 23 pairs, because in each pair you have one coming from your mom, one coming from your dad. So out of those 23 pairs of chromosomes, the first 22 pairs are known as autosomes. So let me write it down. So they're known as autosomes. And these chromosomes code for everything and anything other than defining whether you're male or female. So they code for all traits apart from being male or female. Right, so they're called autosomes. And you've got then this one pair which is called sex chromosomes. So we only have one pair of sex chromosomes, and you can guess what they determine, I hope, right? So they code specifically for whether you're a male or a female. So that's what they determine. So you have obviously two options here. So if you're female, then that pair of sex chromosomes is known as XX sex chromosomes. And if you are a male, then you have XY sex chromosomes. Now, why is that? Why are they known as XX and XY? Well, because in a female, sex chromosomes have got this kind of traditional appearance of what we associate with chromosomes. So I've drawn those sort of pictures already, right? So they look a little bit like letter X. So you have two X-shaped chromosomes as your pair of sex chromosomes, whereas in a male, one of them is that kind of normal, if you like, in inverted commas, traditional chromosome shape. But then the second chromosome is a little bit different. So it's got a little bit of sort of leg missing, if you like, right? So somebody, whoever was kind of naming those chromosomes initially, they looked through the lens of a microscope, saw this pair and thought, well, what is the kind of the name I'm going to give them? So they settled on XX and XY. Okay, so these are some of the kind of key bits of terminology which you do need to be familiar with and be comfortable using when talking about cell division. Now, I've also included a link to Khan Academy video which goes through the same topic, and he kind of looks at very similar pictures, but he actually goes into a bit more detail. So do feel free to also watch that Khan Academy video, and I'll be back with you hopefully quite shortly with the next video. Thank you very much.